Okay, good afternoon, everybody, and welcome back as we make our way through our discussion about genetics. Today, in particular, we're going to be talking about human genetics, and we'll just consider several factors that impact on our study of human genetics, see some of the diseases and the genetic causes, and how we track them in human beings. We ended up last time, we ended up talking about polygenic inheritance. Polygenic inheritance of characters such as height, skin color, these things which are determined by many different genes, but all contributing to a particular phenotype. So we also need to think about things like disease, where there may be, which may have many contributing factors, each of which may have their own set of genes associated with them. I'll give you an example. That's a complex sentence, but I'll give you an example. Let's think about heart disease, for example. Heart disease is commonly caused by blocking of arteries on the coronary arteries, for example, because fats are laid down inside the lumen of the coronary arteries and end up creating a blockage on their own, or else end up encouraging formation of a blood clot which blocks the coronary artery and causes a heart attack. So that is the basic cause. But when we think about the genetic contributions towards this, they can be extremely complex. First of all, it is noted virtually anybody can get a heart attack without any family history of heart disease, etc. But the chances of suffering heart disease are enormously increased if there is a genetic history. So there are definitely genetic contributions to predisposition for heart disease, but these can be multifactorial. For example, somebody with a predisposition to heart disease may have differences in the structure of coronary arteries. They may have differences in the structure of the lining of the coronary arteries. They may have genetic predisposition to laying down fats in the walls of the artery. They may have genetic predisposition to processing lipids in such a way that the lipids are more easily laid down. All sorts of genetic contributions all sorts of genetic features may contribute to a predisposition to the disease. The genetic characters are not necessarily all related to one another. Think, for example, the fact that the person has slight differences in the structure of the coronary arteries is quite unrelated to the way that they process lipids. And yet, put those two facts together and you provide a strong predisposition in that person to having a heart attack. That is what the difference between multifactorial contributions and polygenic considerations. So this multifactorial approach applies to many different diseases. Heart disease we discussed, cancer, alcoholism, mental illnesses. All of these things have multifactorial cause, not only genes, but also lifestyle and diet, and all sorts of other contributing features may coalesce to predispose a person to one of these diseases. In human beings, it is often very, very difficult to sort out what is genetic predisposition and what is due to lifestyle, because lifestyle can actually overwhelm the genetic function. A person with perfectly healthy lipid metabolism, for example, can push themselves to a point where they begin to lay down lipids in their heart vessels simply because they eat so much fat. And this is, for us in modern times, this is a tremendous problem. We are, as human beings, we are, for example, predisposing ourselves to illness very often purely from lifestyle. We're laying that over genetic predisposition. And so, for example, in Western societies, all of these heart disease, cancer, alcoholism, these diabetes, all of these diseases, which definitely have a genetic component, are in fact being enormously increased because of lifestyle adoptions by the population. Sorry, I meant to do this. There we go. So, in other words, you may have a certain genetic predisposition. You may have a certain genotype, but lifestyle can have a huge effect on the phenotype, on what actually happens or how that person functions in the world. Now, as far as the genotype is concerned, there's not, you would realize that there's not a lot that you can do about the genotype. That is your inheritance. That is your inheritance from your mother and your father, but from their parents as well. When we think about meiosis taking place, what we're actually watching is a reshuffling of genes which were inherited from your grandparents, effectively. So there's not a lot that you can do about the genotype that you inherit, but there may be a lot that you can, knowing what the genotype is, there may be a lot that can be done in modifying the lifestyle so that disease, as far as possible, is avoided. The circumstances whereby the disease is created in the phenotype may be avoided. But to do that, you need to know what the genotype is likely to be. And that often in human beings is extremely difficult to do. But if there is a particular concern, for example, this is now commonly done for breast cancer, where women feel that they have a familial history of breast cancer. There are definitely certain breast cancers which are strongly genetically determined. And by examining the genealogy of a patient, a genetic counselor can look back, see what is the proportion of women in a family who have suffered from breast cancer, and what does this say about somebody sitting in front of them? What is the likelihood? And what the genetic counselor will do will state a risk for that particular disease. And if it is also possible in some cases, breast cancer is only one example. There are many other genetically inherited diseases which a genealogy can be examined for. And advice can then be given about changes in lifestyle or even medical procedures which could be adopted in order to avoid the disease that is being predisposed for. So, for example, in the case of some breast cancers, not all breast cancers, but in the case of some familial breast cancers, it might be recommended that the woman have a prophylactic mastectomy. This is indeed done to avoid the development of metastatic breast cancer. But there are also sets of biochemical tests which can be administered to see if a genetically inherited disorder is actually producing phenotypic signs which can be determined by biochemical testing. This may also very frequently be done either on a fetus in utero or on newborns to test whether or not a particular genetically inherited disease is present in that infant. So quite apart from that, we also need to realize that many of the human diseases are carried in a recessive form, and they do not appear in many of the people who actually carry a gene for them, because they're inherited in a recessive form. They only have it on, for instance, on one of the chromosomes. Those people we refer to as being carriers, and identifying them is very, very important, because the character is actually hidden. And if two carriers were to meet and have a child, then there is a likelihood that a child could be born homozygous recessive and express the disease. And so a lot of effort has been gone into identifying carriers of a particular disease, so that they know, they understand, and if when they marry, when they have a child with someone, they are able to assess the risk of producing a child which might have the disorder. It's just a way for people to be informed about their genetic status, about their genotype, because they're not expressing it in the phenotype. This raises a lot of ethical issues, however, because some people just don't want to know. So do people have a responsibility? Do they have an ethical responsibility to know and explain their genotype? It's a very difficult question. In addition, there are disorders which, some people think, there are conditions which some people think are disorders, which other people do not think are disorders. And it's merely, I'll give you an example. Deafness can be inherited, but a person who's deaf might not regard being deaf as a disorder. They manage perfectly well. They manage in the world just as well as anybody else. They do not regard having a child who is deaf as being giving birth to a child with a disorder. Other people might disagree with that. These issues of human genetics and human inherited characters become extremely sensitive. And any move towards cleansing the human population or cleansing a familial line of particular genes begins to sound highly unethical, and is in fact what we call eugenics. Eugenics is an attempt to move the human genotype towards some sort of ideal. And the problem is, who decides what that ideal is? Is that ideal highly intelligent? Is that ideal a certain color? Does that ideal have a certain sexual preference, or sexual identity rather? What all of these things are subjective, and that is a great difficulty with human genetics. It is very often a judgment call rather than a scientific call. So here are some of the ways in which we can test, for example, a fetus. This is done particularly if it's believed that there is a risk to infant and to mother of some particular condition. For example, we can sample fluid from the amnion, which is one of the coverings around the fetus in the uterus. The amnion is one covering, there is another underneath, and between them there is fluid. And that is the amniotic fluid, and that can be sampled. Put a needle through the wall of the uterus and take a sample. It usually has fetal cells circulating in it, and those the genotype can be determined. Very often a karyotype, that is an outlay of all of the chromosomes, can be done to check for chromosomal abnormalities. And there are various other genetic disorders that can be tested for. Another way, now that is called amniocentesis, sampling the fluid, amniocentesis. That carries a risk. It carries a risk to the mother and to the fetus. And it can result, for example, in miscarriage of the fetus. There is another way to sample, and that is to do chorionic villus sampling. Now, in chorionic villus sampling, part of another membrane, the chorion, which is sampled, but it is sampled from the placenta. You may not know, the placenta is this body of tissue anchored against the uterus wall, which communicates with the mother's tissue. They are quite separate from one another. And the placenta is in fact fetal tissue. So if you sample from the placenta, you're sampling fetal tissue. And from that, there are various genetic disorders, genetic status can be determined. Then there are all sorts of other things that are done routinely now. Ultrasound is done to check on the morphology of the fetus, to check it looks okay and everything else. I'll just mention that here, indeed, it used to be, for example, that amniocentesis was done on women who were over 40 years old to check to see if there was a trisomy on chromosome 21. That is, did the fetus have Down syndrome or not? Trisomy 21 leads to Down syndrome with a characteristic phenotype. And it used to be routine that if a woman had trisomy 21 of a fetus, she was offered a termination of the pregnancy. That was why they were doing the amniocentesis, was to check for. Well, we need to think about that. In the 1980s and whatever, it was considered almost routine that this would be done if a child was trisomy 21. The pregnancy would be terminated. But you know what? How many of you know somebody who has Down syndrome? They are marvelous human beings. And it becomes a real question as to whether this amniocentesis is actually that good an idea. It is not a decision that is a decision which the mother has to make as to whether the pregnancy should be terminated or not. But I will tell you that it's becoming less and less common for amniocentesis to be done with an eye to terminating a pregnancy. The mother may need to know, may be good for them to know that this is going to be a Down syndrome infant, because there are all sorts of plans that might need to be put into place for dealing with it. But again, as soon as you start talking about human genetics and about dealing with this kind of problem, you are wandering into a very sensitive ethical region. And it must almost always be borne in mind. Fine, if you find a genetic disorder in an infant or an adult or anybody else, that is one thing to know that it is there. But what you are going to do about it is an entirely different question that may lie outside of the medical and scientific realm. There are many disorders, however, many genetic disorders, which, if they're identified early, can actually be dealt with in one way or another. And an example of this is this disease called phenylketonuria, which is an inherited, it's a recessively inherited disorder, and it is more common in Semitic peoples, Jewish people, Arab. It's for some reason tends to be more common there. That doesn't say it doesn't occur elsewhere. It does, but it's much more common in those populations. But it's relatively, as far as genetic disorders go, it's got relatively high prevalence, 1 in 10,000 to 15,000 births. Doesn't sound that much, but as genetic disorders go, that's quite high. And it's as it's recessively inherited, the gene may be present in both parents and not be apparent to them, but their child will be at risk. And it can be, as long as they're in utero, the child is fine. But after the child is born, they fail to metabolize lipids properly. And the lipids begin to accumulate. The child initially is born healthy. The lipids begin to accumulate in the brain tissue, and they go into a decline and ultimately die. But it can be tested for at birth. And if they have phenylketonuria, then they are put onto a special diet. And on this diet, they can survive perfectly well for the rest of their lives. So it's very important that they be tested at birth, and phenylketonuria is one of the things which is routinely tested for in infants as they're born. But there are many others, and the list is growing. There are numerous biochemical tests which can be administered to newborns in order to modify their upbringing if need be. Now, in general, you'd think by this time we'd know a huge amount about human genetics. In fact, human genetics is still an area of obscurity in many ways. And this is for some very good reasons. First of all, we have relatively few offspring per parent. And so your sample size is small. It's not like dealing with the pea plants where you could breed thousands and look at the data and get a good statistical spread. Human beings, we seldom reproduce in actual fact. Second of all, our generation time is very, very long. So if you want to trace genetic traits through generations, you've got to wait an awfully long time. And as the genetic scientist's generation time is as long as his subjects is going to be dead before the great-grandchildren that he was interested in make their appearance. So that makes studying very difficult. The other thing is we don't breed people deliberately with one another. And that is unacceptable. We don't breed human beings as an experiment. We have to just follow what is happening naturally. And I'll give you one other thing which is little talked about, but which every genetic scientist who is interested in this kind of familial inheritance will tell you. And that is quite often human beings don't behave the way they are supposed to behave. And so, for example, it is very frequent when people are following a particular family or traits through a particular family, they will suddenly find that there are offspring whose parentage is obscure, which is, a family secret or a secret of one member of the family, which they have kept. And this is actually incredibly common And because we have so few offspring per family, and because our generation time is so long, this can, just one of these instances in a family can throw off all of the patterns of inheritance that are observed. So that is something that needs to be borne in mind. Human beings are naughty things, okay, and they sometimes do things which a scientist would rather that they didn't do, because it throws the numbers off. However, it has to be said that when we look at an individual gene, if we look at the behavior of a gene, we can often see simple Mendelian genetics applies, that it's still the basic principles of Mendelian genetics endure. It's just that it may be very, very complex, and very often the issue of polygenic and inheritance raises its head, and so do things like epistasis and all of the complicating features. The standard way for a genealogist, or rather a counselor or whatever, to examine genetic inheritance in human beings is to draw up a pedigree, to look through the family tree and to try to identify who showed certain traits, because by looking at traits, we can actually work out sometimes what the genotypes of parents, grandparents, etc. were. So, for example, let's have a look here. These are just simple things. If we were doing a genetics lab, we would do some of these. These are pedigrees here, and they show parents married with children. So here's how it works. Squares here are male. The circles are female. If the person has a particular trait that we're interested in, then they are colored. They are colored in like this. When a male mates with a female, that is there. And we show, by convention, we show all the offspring with the oldest one here and the youngest one here. So we're going to examine now for a simple, harmless trait, and that's the widow's peak. Can you see here this lady? She has this point there. Okay, there. This is a sister. Her sister has no point. That's called the widow's peak, and it is genetically determined. It is by a simple inheritance, single allele. And so she doesn't have it. She has it because she's female, so she's circled. She has the trait, so she's colored in there. She's also female, but she does not have the trait. Okay, let's go back. Let's have a look through the pedigree of these two sisters. Here are their parents. There's their father there, and he has the trait. There's the mother there, and she also has the trait there. Let's think about this in Mendelian terms. Okay, we're looking at a trait. We don't know yet whether it's dominant or recessive, but what we can say is that these have to be heterozygous, right? Because look at the outcome here. They have to be heterozygous because the trait has made its appearance in both allelic forms. Here she has it, she does not. Okay, let's go right the way back. Let's go back to the grandparents. Here is the grandfather, here is the grandmother, here is grandfather, here's grandmother. Does not have the widow's peak. Did have the widow's peak. This grandfather had the widow's peak. This one did not have this grandmother did not have the widow's peak. Look at the distribution here, 50-50, about, has there, does not have there. Has there, does not have there. There's only one way to explain this inheritance. We need to determine whether or not this widow's peak is a dominant trait, or if it is a recessive trait. And the answer is that must be a dominant trait. It has to be dominant. Look here, if she here is either homozygous or heterozygous. She is homozygous and does not have the trait. Let's go all the way back. We can work it all out. Here's the grandfather here is heterozygous. The grandmother here is homozygous. What possible combinations of genes could they produce? Big W, little w, little w, little w. You could work out a Punnett square if you wanted and work out what a proportion should be. Remember though, human beings have very, very few offspring. So they're not likely to fit in, statistically fit. But here we have the range of possibilities of genotypes. Heterozygous, heterozygous, homozygous, and homozygous. It looks like this the recessive trait is no widow's peak. The widow's peak is produced either by in the homozygous condition, WW, or in the heterozygous. So if we ask the question, is that widow's peak dominant or a recessive trait? It must be dominant. It is dominant because it appears equally in the homozygous and in the heterozygous. So this is a simple way of just going back, working out what the likely genotypes are. Notice we can't definitively say what hers is. It could either be homozygous or heterozygous. Look here. These are both heterozygous, but they could produce a homozygote, or they can produce a heterozygote, or they can produce a homozygote recessive. A simple trait. Have a look here. Here's another one. You can work this one out for yourselves. Try to work through this and work out. Here is the pedigree here. Here are the genotypes. And now, if you just work through where the appearance here is this trait, a homozygous, a dominant or a recessive trait. For tasting, this particular substance is called PTC. And if you can taste PTC, you can see the look on her face. It tastes bitter. If you can't taste it, you can't taste anything at all. Again, have a look through here. Here we have two heterozygotes who cannot taste. Oh, sorry, who can taste. Yes, the cannot taste is the trait we're looking for. Sorry, cannot taste is the trait we're looking for. So this is a taster. She's a taster. She is a non-taster. So is the inability to taste a dominant or a recessive trait here? It is a recessive trait there. Here are our two heterozygotes, and in the homozygote or in the heterozygote here, she can taste. So the inability to taste is the recessive trait there. So that's the way that you do it. You look at all of the ancestry as far as back as you can go, see who has it and who doesn't, and you can begin to work out the pattern of its inheritance. Here is an explanation for you here. I'm not going to do this now, because these things just don't work on Zoom, unfortunately. Can't hear anything. So many genetic disorders are inherited as a recessive. And the reason for that is if they were inherited as dominant traits, it would be extremely damaging to the population. They are extremely damaging if they're inherited as a dominant trait, and they disappear from the population very soon because those people simply either they don't survive or they don't reproduce. But many other disorders are inherited as recessive traits and only make their appearance on occasion when two people mate who both carry the recessive trait. So they are carriers, they're heterozygous, for example. The dominant trait is the healthy trait, the recessive trait is the disorder. And you have two heterozygotes who mate, produce a homozygous recessive that expresses the disorder. They're very huge amount. There's some are very mild, others life-threatening. There are some complications to this process, however. And so, for example, even if it's life-threatening, it may only make its appearance in the adult. Juveniles may survive, and it only appears in the adult. Things like this. The classic inheritance is, as I've said, that these recessively inherited disorders show up in individuals who are homozygous for the allele. Those who carry the gene in the heterozygous genotype are called carriers. They're phenotypically normal, and that is a problem. And this is one of the reasons for going to a pedigree like this and tracing back to see who in the pedigree has it, and then determining what is the likelihood that parents are heterozygous for that trait, and what is the likelihood then that they might themselves produce a child. And here's a classic example. This is if you want to think about it as a disorder, I guess you can, but this is the characteristic of albinism. And people with albinism do not lay down any melanin in their epithelia. So their skin is extremely pale, and they usually have blue eyes because blue eyes are eyes which lack pigment. So this is inherited in the recessive. And here we have parents. Each of them have normal phenotype, dark skin, but these are heterozygotes, big A, little a, big A, little a. This recessive gene here is the gene for albinism. So when we look at their, if we were to draw up a Punnett square for the likelihood that they will actually produce albino children, what genes can they produce? Big A, little a, big A, little a, big A, little a, there. So do the Punnett square, and you get homozygous dominant there, AA, normal. That's the normal laying down of melanin. This here, heterozygous, AA, big A, little a, big A, little a. These are carriers, but the phenotype is normal. This is a dominant. That is recessive. This is enough for them to have normal colored skin. But the phenotype is 3:1. It's obeying simple Mendelian genetics. This here is double homozygous recessive, little a, and that is the combination which will produce albinism as the phenotype. And here it is here. The albino people do have some problems. They do have eye problems associated with the phenotype. And they're also very prone to skin cancers. This should not surprise us because, in fact, the melanin is a protective mechanism which protects the skin from ultraviolet light. But otherwise, it's a relatively harmless inheritance. There's just a couple of things that people who are albino have to be aware of and be careful of, but otherwise it's completely, the rest is completely normal. So this is a typical recessive inheritance. Is often also, if you have somebody who is albino, who marries somebody with a normal phenotype, then it goes again, classic inheritance by Mendelian principles. Their offspring will all be heterozygous. And the albino recessive gene will reappear in the F2 generation effectively. If one of those children, if one of the heterozygotes is to marry another heterozygote, then it has a 3-to-1 chance of, sorry, a 1 in 3-4 chance of appearing in the pedigree. The albinism is a trait which is preserved in populations because it is relatively harmless. And people who are albino go on to full adulthood, and they themselves marry and have children, etc., etc. So it is actually preserved in the population. If associated with albinism, you had some really serious health disorder, it would be likely that the gene would move out of the population, because anybody severely affected by an illness or something associated with would tend to reproduce less. Their chances of reproducing are lower. And the gene tends to move out of the population. This is one of the reasons, by the way, that we believe that most societies have taboos about family members marrying, because you'll realize that these recessive genes move in a familial way. They're inherited within families. And if members of the same family married with one another, they are much more likely to produce these homozygous recessive offspring. And if a carrier moves out into the general population, their chances of mating with another heterozygous are much lower than they are if they marry within their family. And there is different societies have different conventions for how far away you can marry. The normal for most societies, cousin is the closest that you can marry without societal taboo, and that is probably one of the reasons why. So here is another disease which is maintained in our population and is also a recessive, but this is one of the commonest of the really serious recessively inherited genetic diseases. And this is cystic fibrosis. And it's really quite common in terms of genetic inherited disorders. 1 in every 2,500, much more common amongst European Caucasian population than in any others. It can make its appearance in other populations, but it's more common. And this, you'll remember, we talked about it before. This is a pleiotropic gene because it has effects on many aspects of the phenotype. It has effects on many aspects of human physiology. It is results in a defective transport of the chloride ion. The chloride ion transport channels are defective, and that is those lie in the cell membrane, and they it leads to a buildup of salts outside of the cell. And this can have profound effects on the functioning of cells and on the functioning of the whole organism. And it's the principal character of the phenotype is that in cystic fibrosis, the mucus of the lungs is extremely thick and sticky, and the cilia cannot move it. And as a result, debris and bacteria, etc., tend to accumulate in the mucus, and the mucus accumulates around the alveoli and ends up causing lung dysfunction, ultimately lung scarring and everything. But it also affects the function of the kidneys. It also affects the absorption of the nutrients in the gut. It affects the sweat. It affects many different aspects of human physiology. And this is a disease even within memory. Cystic fibrosis was usually fatal before the age of 5, 6 or so. But nowadays, people live for very, very much longer. Treatment with antibiotics, for example, drug treatments to liquefy the mucus, and various other things. Nowadays, many people with cystic fibrosis now survive into their 40s. But here is another ethical dilemma, and that is that now people with cystic fibrosis very frequently survive into reproductive age where they did not even 30 years ago. And so now the genetic counseling becomes extremely important because they are now able to perpetuate that gene where and they're able to propagate the gene rather into the population. So this is something that they need to think about very, very carefully if they are themselves going to reproduce. Here's another one. This is sickle cell disease. Sickle cell disease is actually quite common, particularly in the African American population. And there is a question to be asked: why is this so common? It can be extremely serious disease. We'll try and answer that question in a minute. So sickle cell disease caused by the substitution of a single amino acid. A valine goes in where a glycine should be in the polypeptide that is going to go on to make hemoglobin. And because of that, the nature of the hemoglobin changes, and it results in this disease of sickle cell anemia. The effects of sickle cell anemia vary tremendously. Some people tolerate it. So other people, it actually becomes lethal because they end up having organ damage and all sorts of things. Here's the basic way in which it works. I think we talked about this before, but it bears repeating. So it's a variant on a single gene. So we have alleles which are inherited either in the homozygous or in the heterozygous form. And in the homozygous form, now normally blood corpuscles, the hemoglobin actually does change shape inside the red blood corpuscle as it takes up oxygen and gives up oxygen. But normally the hemoglobin and the red blood corpuscle itself are very pliable. They can fit through the little fine capillaries of the blood circulatory system quite easily. They slip through because they are able to deform and then adopt their normal form outside the capillaries and flow. But unfortunately, in sickle cell disease, what happens is that the hemoglobin is actually altered. And when it goes into very low oxygen conditions, where it would normally drop its oxygen and change shape and then be conducted back to the lungs for reoxygenation, Now in sickle cell, the hemoglobin start to stick together like this, and they form fibers, which are almost contractile. They cause the blood corpuscle to become rigid and bent in shape like this. That's where it gets its name from, sickle cell. These fibers and the deformation of the red blood corpuscle mean that they can't get through the capillaries. And they start to block up the capillaries. They set up an inflammatory condition in the capillaries. The immune system begins to react against them. And that is what causes the symptoms of the disease. In a homozygous condition, all of the red blood corpuscles are liable to this. In the heterozygous condition, however, they have one copy of the sickle cell allele, one copy of the normal allele, and they are able to, half of their hemoglobin is normal, and half of their red blood corpuscles are normal. And that is usually enough to keep them going. People who are heterozygous show a variety of different disorders. They may be very mild, they can be relatively severe, they may be have it only episodically as an acute episode, but then recover. People who are homozygous are much more seriously affected. So the question is, now, this is a recessive, but why is it being maintained in the population? This is very, very serious, and people may even die young or die early, whatever. How is this being maintained in the population overall? Well, the answer, we believe, lies in the fact that the heterozygous condition is actually advantageous. It is advantageous where malaria is endemic, because malaria parasites cannot infect the sickle cell blood corpuscle. And in fact, the overall population of blood corpuscles does not support a healthy malaria population, healthy for the malaria. People with sickle cell heterozygosity are at an advantage in places, for example, like West Africa, where many of our African American population originated. And they have brought this trait with them through all of the ages. The trait has been maintained and it makes its appearance as sickle cell. It is a particular problem when two people with sickle cell who are heterozygous for sickle cell marry and produce offspring who are homozygous. That is when it becomes a particular problem. But I must tell you, I think I told you this before, but I can tell you right now that for most of you in this class, you will live to see a cure for sickle cell anemia. I can say that with absolute certainty, because sickle cell anemia is one of the diseases which is top of the list for genetic correction, genetic engineering correction. And that will certainly be within your lifetimes. Okay, so not all disorders in the human population are inherited as a recessive. Again, I remind you about something I showed you earlier, polydactyly. Remember the guy with 5 fingers? Dominant trait can be extremely rare in a population. Just because it's dominant doesn't need to say it's common. And there are some disorders that are caused by a dominant allele inheritance, and are one of the classic ones. Well, first of all, I would say dominant alleles called lethal disease are rare, very, very rare, because they are quickly eliminated from the population if they have that serious an effect, and it is dominant. Remember, it's going to appear in heterozygotes and in homozygotes. If it is a serious illness or lethal disease, it is quickly going to be eliminated from the population, especially if it's inherited as a dominant allele. But other things, we wouldn't really say they're disorders, is a bit pejorative. there are things which move through the human population, which are inherited in the dominant form, because they're not lethal. They're not really a disorder. And the classic one is dwarfism, achondroplasia. Achondroplasic dwarfism is inherited as a dominant. And have a look here, and you'll see we can immediately tell. Here's somebody who is heterozygous here for dominant, and this is the dwarf phenotype. Somebody who was dwarf married somebody of normal height, and in this pedigree would be designated little d, little d. The dwarf phenotype would be D, large, big D there, big D, little d. Certainly, two dwarfs can marry and do and will have dwarf or normal children. But here in this pedigree here, for this particular gentleman, here is the father, dwarf, heterozygote, big D, little d. Mother, normal height, little d, little d. And what the possibilities are: big D, little d for sperm, little d for the eggs. And in this case, what do we have? We would have a 50-50 likelihood of producing the dwarf phenotype: big D, little d, big D, little d, dwarf, dwarf, normal, normal. This should remind you of something. This looks like a test cross, right? We've crossed a homozygous recessive with a heterozygote, and this is what we get. We get a 50-50 distribution. And this is a gentleman here. He's actually quite famous, and he is an orthopedic surgeon. And there are some problems associated with achondroplasia, and in particular, it is deformity of the long bones, which can produce tremendous problems, especially later in life. And he's a very famous orthopedic surgeon, and he works specifically on achondroplasic syndrome and corrects bone deformities and things like that. Okay, there are some dominantly inherited, very serious conditions which persist because they only make their appearance late in life. And one of these is this disease, Huntington's syndrome. It's sometimes called Huntington's chorea. Chorea refers to shaking. And people with Huntington's chorea, that may be one of the symptoms of the phenotype, is that they have a palsy, a shaking. In any case, Huntington's disease is a degenerative disease. It's a dreadful disease. It's a degenerative disease of the whole nervous system, but particularly of the brain. And it has no real effect until people are 35, 40 years of age, and then they begin to decline, and it is irreversible and fatal, ultimately fatal, and a very nasty disease indeed. It is inherited in the classic Mendelian dominant way, but formerly, the only way of trying to predict was to look at a pedigree and see which of the parents, which of the grandparents had manifested Huntington's disease in their later life, and then try to predict. And children basically had a 50-50 chance of either expressing it or not expressing it. And but nowadays there is a test. There are tests for the Huntington's allele in the genome. And again, this raises a tremendous question. If people have a history of Huntington's disease in their family, do they choose to be tested for the allele or not? well, knowing that there's actually nothing that can be done about the onset of disease, many people choose not to be tested. They rather just wait and see what happens, rather than live their lives in terror. Others do choose to do so, especially if they want to marry and have children. They want to know, are they carriers, that are they going to pass this gene on to their children, etc. It's a very delicate decision, because this is such a terrible disease, and its outcome currently is a fatal outcome. I'll just mention one very famous instance, and that is the singer Woody Guthrie. And Woody Guthrie, he had Huntington's disease. His sister also died of Huntington's disease. But apparently his most famous child, Arlo Guthrie, does not have the disease. So one of the things that we do need to bear in mind very strongly, especially when we're talking about human genetics, I think, and we're talking about that multifactorial inheritance, which applies to many, many, many of the human conditions. We need to remember that the phenotype is not just one character, right? The phenotype is a whole lot of characters expressed in the organism, and the phenotype is the functional organism. That is the way that the organism has to interact with the environment. And what we observe is, and in the organism, is an interaction between the genotype and its environmental history, how that genotype interacts with the environment. When we're talking about multifactorial disease, we were talking about all of the genetic things, well, structure of the vessels, how the person metabolizes lipids, etc., etc. And then observing, was there genetic disease? Was there a heart disease phenotype that was observed? Well, we must always bear in mind this interaction, especially in human beings, between genotype and environment, between what we're given and what we do with it. Just as a simple example, have a look here. These are two hydrangea plants. They're actually clones. They're genetically identical plants, but one of them grown in soil which is alkaline, one of them grown in soil which is acidic, and they are completely different colors. We would say that these are totally different plants. Well, they're not. They're clones of one another, but the phenotype is different. This is the red phenotype. This is the blue phenotype. The organism interacts with its environment to produce what we observe as the phenotype. And I think that with that, for this section, that is it. Okay, have a good afternoon. I will be putting up a new assignment this week for you. And also, please note that I put up the answer sheets to your tests for you to begin going through your tests and studying already. Please start studying for your final, which is coming up very soon. You have one test coming up on November 23rd on unexamined material, but then don't forget that your final, you have comprehensive as well.